Greetings and welcome to a video to talk about cell signaling. Cell signaling is one of the key aspects of making sure that our immune system is able to recognize friend from foe. So in this video you will learn about two different cell signaling molecules, the MHC1 and the MHC2. And these things are going to help us identify in the body whether or not we have a friend or a foe. Okay, so when we're talking about cell signaling, we need to remember that cells don't have ears, they don't have eyes, but they do have ways to communicate between each other. And what cells typically do will send out some form of signal, and there is some sort of receptor. So within our cells, the cells have the ability to signal each other by putting structural surfaces, proteins, on the outside of their cell. What this looks like is something like this, or something like this. But before we can really understand what the cell is doing in its attempt to signal, we need to take a quick moment and think about other ways that signaling happens in the world when we don't have the ability to use our ears. Okay, let's take a minute to consider MHC1 signaling, the Major Histocompatibility marker that is utilized by cells across a body to represent that they are a part of a collective or a part of a whole. So we're going to think of MHC1 as the way that cells typically signal that they belong, to create a sense of belonging amongst the body and to help the body represent and understand what is self versus not self. So as we think about major histocompatibility markers, we think about this protein, we need to recognize a couple of features once again. The message that we expect from an MHC1 protein compatibility marker, a histocompatibility complex, is that the cell presenting the MHC1 marker is relaying to other cells in a multicellular organism that it belongs, that it is self and is part of the community as a whole. So, as a key point, usually, if a cell is representing itself with an MHC1 marker, it's communicating to other cells that it is a cell that belongs. Now, occasionally, a cell that belongs might have a situation arise that makes it a potential threat. This could be in a situation where perhaps the cell has gotten cancerous or tumor, the cell cycle is elevated, or abnormalities are detected in a cell. This can also represent anytime we get what's called an intracellular pathogen. These are most commonly viruses, but there are a few protozoans and bacteria in the world that also like to make their home nestled inside of the host cell. So let's take a quick moment to look at how usually MHC1 is used as a marker. Okay, so remember, we're going to talk about what is usually the case in the vast majority of the time. And so what we've done now is drawn a cell to represent the various cells across the body that are going to use some form of signal to say that they belong. And in this cell, we're going to draw ourselves a nucleus and a little bit of endoplasmic reticulum around it. But we're also going to draw a couple of little dots in here to represent the ribosomes. And you may remember that ribosomes are also going to be found out in cytoplasm. They typically have two different pieces associated with them. And the ribosomes are going to be a key component of a process we call transcription and translation. So what typically happens is in the nucleus, in the center of the cell, there are codes for proteins of belonging. So the genetic material is going to have some information that says we belong, and this is also going to include some information that is going to make my MHC1 protein. Now all of this, if I zoom in, is going to be a component of that double-stranded DNA molecule. And in that DNA molecule, we have these different genes. And so if I zoom into the gene, I'm going to be able to read the code of the gene. And what we're saying is we have one of these that tells us how to make an MHC1. We have one of these that tells us how to make an MHC2 as well, but there are also other proteins that the cell is making throughout the course of the day to run its day-to-day operations. But in the light of signaling, what's going to happen is through the process of transcription, we're going to make a copy of the gene from the genetic material on the inside of the nucleus. And that copy of a gene is going to head out to a ribosome, and it's going to feed in kind of like a ticker tape through the center of the ribosome. Now, simultaneously, other ribosomes may be working on other genes. So here's my little ticker tape coming through of the gene that's going to code for the histocompatibility complex versus proteins that are self proteins generated from the inside of the cell. So through the process of translation, we take the information that was created or sent from the nucleus. We utilize a ribosome. So these guys indicate my ribosomes. And you guys may remember eukaryotes are 80S, prokaryotes are 70S. And that's a flashback to an idea from a general biology class. So these ribosomes are going to make proteins. And so as this is being sent through the cell, on the far side, we're going to make an MHC1 protein. And the MHC1 proteins are oftentimes illustrated a bit like an X. This is not to be confused with a chromosome, but they have a middle area. They have an area where we can bind an antigen. And we have another area that serves as the anchor of the protein to help us embed it into the plasma membrane. So once we make this protein, we're going to take this protein and we're going to put it into the surface of the cell. So remember, one end is the anchor that's going to help hold it into the plasma membrane, and the other area I'm going to be able to create an antigen. But what antigen is going to go on the surface? Well, this other ribosome that was creating an antigen, in this case we'll make it a blue ball. This antigen is a self antigen. This is something the cell made to signal to other cells that I am making the same types of antigens you are. I am you, we belong, we're all in the body together. And so the MHC1 flagpole, that marker, will present this antigen on the surface of its cell to remind everything else. So in this picture that I just drew, I very intentionally drew the cell in blue and used the blue ball as an indicator of self so that other cells who are doing the same thing can be headed around and they can all be wearing their ball antigen that indicates that I am self. So all cells across the body are going to be running through this process. All cells across the body are going to be creating these flagpole MHC1, this marker to indicate, it's kind of like a flagpole. And that's going to be able to allow cells to recognize each other as communally all belonging to the same organism. So this is kind of an interesting idea, right? If I think about this, here's the bottom of my cell, and I can kind of think about these MHC1 markers as a type of a flagpole. Should have drawn that here in this color. We're going to think about it as a flagpole. And then off the surface of that flagpole is going to be the flag that carries the antigen that allows other cells to recognize each other if they are all communally part of the same body. Okay. So that was a key idea to think about in reference to the fact that this is a we belong kind of situation. And the way that this cell signal, this major histocompatibility signal is created is different than what we see in MHC2 processes. So now we're going to switch screens and we're going to consider what's happening with the MHC2 molecule as it goes through a processing and how we create that flagpole and what sort of message that is going to relay to my body, to my other cells. Body to my other cells. Let's take a moment to consider what MHC2 as a signaling molecule is and how it's used to help coordinate an immune system response within the body. This is a key component to signaling between cells to help educate the cells that are able to attack what they should be attacking. So this involves a signaling molecule that's known as an MHC2 molecule. This molecule, it MHC stands for Major Histocompatibility. So this is a way for cells to be able to determine and signal what is compatible versus not compatible within the system. When I think about an MHC2 molecule, I think of it as a wanted poster, similar to the ones that you might see on the street for a wanted or missing pet, or perhaps a wanted poster for America's Most Wanted. So up in the corner, you'll see our wanted poster, and this has the evil red ball antigen that I frequently refer to. Now, if you are not familiar with the phagocytosis process, please consider reviewing the phagocytosis video and looking at how the cells are ultimately going to be bringing in foreign material that they can then use to craft their wanted poster. So let's get started and start to think about what happens after an antigen-presenting cell, like a macrophage, manages to degrade an evil or a foreign cell that needs to be removed from a body. If you are familiar with the processes of phagocytosis, you know that it's a 4-step process that ends in digestion of a cell into bits. So at the end of the process of digestion, you have a lysosome and a phagosome that have fused together to create this structure illustrated as the phagolysosome. At the end of digestion, the foreign cell or the evil cell was broken down into pieces and is now destroyed. But we can use those pieces to be able to signal to other cells that there may be an invader and there may be something to be on the lookout for, aka our wanted poster. For a cell to be able to display a wanted poster, another couple of steps, another couple of things need to happen in order to get that signal out on the surface of the cell. The cells that do antigen processing have pre-made MHC2 flagpoles or MHC2 proteins. So what you'll have seen illustrated on the board now is this little X-shaped, I think of them as hot dog buns that have been kind of splayed open on the ends. And this is essentially a flagpole, something that the cell can use to put different things on the surface of the cell in order to present the antigen of the thing that it just made and to create the wanted poster. So these cells that are processing antigens and are presenting them on the surface will use the process of transcription and translation to pre-make these flagpoles so that they can very quickly get that information up onto the surface of the cell and be able to signal to the other cells in a rapid fashion that they have detected an invader or something that doesn't belong that other cells should be on the lookout for. So, to bring us back to the end stage of digestion, once the phagosome has digested and we have remnants of the cell that was destroyed, the macrophage or antigen presenting cell is going to take an MHC2 marker that it pre-made. It's going to put it into the plasma membrane, and I like to think of the bottom of that hot dog bun kind of splayed open as a way to sort of anchor this flagpole onto the surface. But without something on the tip of the flagpole, there's no message to be sent. So in the case of the phagosome, phagolysosome, we're going to take that MHC2 protein that was produced and we're going to take a remnant of the cell that we digested and we're going to put it onto the surface. So this is the evil red ball antigen. Antigen meaning the thing that is used to signal. And so now our cell has displayed this evil red ball antigen on the MHC2 protein that it pre-made, little flagpole, and essentially what it's doing is through the cell, it's running a flagpole up, and on it, it is putting a flag with the red ball so that other cells will be able to come along and recognize this thing as a wanted poster. This is something that the antigen presenting cells have determined is not a component of the body, not something that needs to be there, something we want to seek and destroy. And the wanted poster, the flag says, hey, any other cell, if you come up and you find this red ball antigen, we want to attack it. We want to kill it and eliminate it. So this is very important in cell signaling to start to pass on the information of this is something that we want to seek and destroy. So let's sum this up. What are the key messages of an MHC2 signaling protein? This is a protein that cells make that are antigen presenting, and they create these ahead of time through the process of transcription and translation to be able to be prepared to rapidly mount a response if a foreign invader or something dangerous is determined. The overall message that the MHC2 signaling histocompatibility marker indicates is that something has been located in the body that is dangerous, whether that's a cancerous cell, whether that's a virus or a bacteria or a fungus. It's something that doesn't belong. And so it displays a wanted poster to signal to the cells that actually have the ability to do something about this, what their specific target ought to be. So think of this maybe as the commander is putting out an all points bulletin, and the Navy SEALs now know exactly what their target is and what they should go and look for based on the wanted poster that they were presented with. Over here, remember, we had MHC1, and MHC1 largely signaled, I am you, I belong, please don't attack me. Now, you may be familiar with the fact that cells like macrophages and dendritic cells are really good at eating things. Make that a little more obvious. Dendritic cells. So these are really good at eating things, but there are also infections that a cell might experience where they get a hostile takeover. So if we think about a hostile takeover, this is common for things like viruses. So if I were to get a virus, I'm going to just draw this as a hexagon structure with a little squiggle to represent the genetic material that this virus might bring. If the virus sneaks into the cell, then the cell is going to have to stop doing the cell's business, and now it has to listen to this new weird warlord, the virus, that's going to go in and take over all control. Now, the cool thing about this is that this cell may have to do what the virus tells it to do, but it can send an SOS. And it does that by taking, right, so it's in here and it's making more copies of viruses, and we're getting all these little bits and pieces. And what'll happen is we can use this MHC1 marker, this flagpole, we can actually put this on the surface of the cell again. Okay, only this time, instead of putting the antigen, instead of putting the thing on the surface that says, this is me, this time he sneaks out and displays that little tiny bit of the virus. And what this does is allow cells to come up and say, okay, wait a minute. If I'm going to communicate with this cell and I find the me antigen, here it is up here, okay, there's my little protein that says, I am a me cell, leave me alone, we all belong, we're happy together. But if the cell gets taken over by the virus, it can use the same flagpole, but a different flag, right? A different message, and it puts a little piece of the virus on the surface, and it changes the message. And the message may say, I am infected. And if this is the case, if other cells in the body circulate around and they discover, oh no, we have an infected cell, what are we going to do? Well, the message sent by an MHC1 that happens to have a foreign protein on it is going to signal to the body, kill me. Kill me now before I inadvertently release more viruses and I infect my brothers and sisters, my adjacent cells. Is this confusing to you? It sure was confusing to me. So I came up with an interesting little analogy to help me understand better what a major histocompatibility complex might be like. So major histocompatibility is a flagpole in this analogy. And Prior to telephones and modern society, in many cases, if we had two ships out on the ocean, the way that they would communicate to each other were by flags. So one boat would have a flag, perhaps of its country, and if the other boat had the same flag, they could see from great distances apart that they were compatible, that they were similar or safe. And in the ocean, if these ships were traveling, they might actually get close together, visit a little bit, pass mail back and forth, maybe trade for water or salt or fish or some of the things that were on board, and then they would go on and continue their journey. What if my flag changes? So, if we had these two ships on the ocean and they were both friendly, but one ship had an illness aboard, they were able to run up a flag that said, hey, we're sick, and this would let the other boat know to keep its distance to make sure that they weren't interacting with each other and potentially sharing that illness. So the MHC is the flagpole, but we can change the flags to have different meanings. Here's another different meaning. Here is our pirate analogy. So right now, both ships are relaying with flags that they are similar, that they are like each other. So as the boats draw close to each other and get closer, it's very possible, and not altogether unheard of, for the boat that's coming closer to then throw a flag up that was the Jolly Roger. And then, now, those ships were close enough that it would be easier to attack, to be able to board, to be able to control or take ownership of the additional ship. So, from a mariner's standpoint, we can think of the Major Histocompatibility complex as a flagpole when and where it's created, but that flagpole is very important to the cell in being able to display different types of flags on their surface. And the flags that we talk about for cells are frequently given the name of an antigen. So different cells can present different antigens, and different antigens are going to have different meanings. Okay, time to sum this up. The big picture, the main concepts that are important to understand about histocompatibility is that cells are utilizing different mechanisms to be able to signal to macrophages, immune system cells, what are the things that belong, what are the things that don't belong, and if something is dangerous versus not dangerous. The key concepts here include two major histocompatibility complexes, Major Histocompatibility Complex 1 and Major Histocompatibility Complex 2. These are proteins. The analogy is a flagpole that is produced by a cell in order to signal other cells about situations that may need to be dealt with. The MHC1 protein, as we learned, is produced during the day-to-day operations of the cell and is not prepared ahead of time, but rather is built as needed. Whereas the MHC2 protein is going to be prepared ahead of time for special use in cells that know that they are going to be doing more signaling than what perhaps your generalized or your regular types of cells are going to be doing. We talked about a couple of messages. So MHC1, largely the message is of belonging. What are the cells that belong? Are they self or not self? However, we said in that we can also relay and signal that there is an infection. So if there's an infection or the cell does not belong, then we're going to see a slightly different response, okay? If you belong, the message is leave me alone. So any of those roving cells, those cells coming out looking to attack and remove cells that don't belong in the body, those are going to be able to come along, read an MHC1 with a self protein, self antigen, and realize everything's A-okay, we're safe. But those same cells have a way to send an SOS, a message out that says, hey, actually, something is wrong internally, and for the greater good of the population, it's probably time for me to die. So anytime we have an infection, cancer, those types of things, your cell is going to be able to signal to the cells that do the cleanup, hey, it's time for me to pack my bags and go. Go ahead and wipe me out. Now, the MHC2, remember, is utilized by a very specific group of cells known as antigen-presenting cells, and their role is to facilitate communication with the immune system. So in this, the message for an MHC2 is, hey, we found something that doesn't belong. This is that wanted poster idea. So we're going to digest the cells that are phagocytic. We'll digest the molecules and the cells and the viruses and the bits and the things that don't belong in the body. And then special groups of them, like a macrophage, have the ability to then present on a flag as an antigen with the MHC2 major histocompatibility, a wanted poster that allows other cells in the body, those immune system cells, to know what the enemy actually looks like and to who to go out and attack. Hopefully, this kind of clears the mud a little bit on the differences between major histocompatibilities and the differences between type 1 and type 2 histocompatibilities with the messages that they're able to relay to other cells. Have a great day.